Metastasis-associated protein Mta2 is a protein that in humans is encoded by the Mta2 gene. Mta2 is the second member of the Mta family of genes. Mta2 protein localizes in the nucleus and is a component of the nucleosome remodeling and the deacetylation complex, NuRD. Similar to the founding family member Mta1, Mta2 functions as a chromatin remodeling factor and regulates gene expression. Mta2 is overexpressed in human cancer and its dysregulated level correlates well with cancer invasiveness and aggressive phenotypes. Mta2 was initially recognized as an Mta1-like 1 gene, named Mta1L1, from a large-scale sequencing of randomly selected clones from human cDNA libraries in 1999. Clues about the role of Mta2 in gene expression came from the association of Mta2 polypeptides in the NuRD complex in a proteomic study. This was followed by targeted cloning of murine Mta2 in 2001. Mta2 is localized on chromosome 11q12q13.1 in human and on 19b in mice. The 8.6 kb long human Mta2 gene contains 20 exons and 7 transcripts inclusive of 3 protein coding transcripts but predicted to code for 2 polypeptides of 688 amino acids and 495 amino acids. The remaining 4 Mta2 transcripts are non-coding RNA transcripts ranging from 532 BP to 627 BP. The murine Mta2 consists of a 3.1 kb protein coding transcript to code a protein of 668 amino acids, and 5 non-coding RNase transcripts, ranging from 620 BP to 839 BP. Amino acid sequence of Mta2 shares 68.2% homology with Mta1's sequence. Mta2 domains include a BAH, bromo-adjacent homology, and ELN2. EGL27 and NTA1 homology, a SANT domain, SWI, ADA2, NCOR, TFIAB, and a GATA-like zinc finger. Mta2 is acetylated at lysine 152 within the BAH domain. This gene encodes a protein that has been identified as a component of NuRD, a nucleosome remodeling deacetylase complex identified in the nucleus of human cells. It shows a very broad expression pattern and is strongly expressed in many tissues. It may represent one member of a small gene family that encode different but related proteins involved either directly or indirectly in transcriptional regulation. Their indirect effects on transcriptional regulation may include chromatin remodeling. Mta2 inhibits estrogen receptor transactivation functions and participates in the development of hormones independent of breast cancer cells. The Mta2 participate in the circadian rhythm through clock BMAL1 complex. Mta2 inhibits the expression of target genes owing to its ability to interact with chromatin remodeling complexes, and modulates pathways involved in cellular functions, including invasion, apoptosis, epithelial to mesenchymal transition, and growth of normal and cancer cells. Expression of Mta2 is stimulated by sp1 transcription factor and repressed by chyso. Growth regulatory activity of Mta2 is modulated through its acetylation by histone acetylase P312. The expression of Mta2 is inhibited by the Rho-GDIA in breast cancer cells and by human beta defensins in colon cancer cells. MicroRNAs 146A and miR34 also regulate the levels of Mta2 mRNA through post-transcriptional mechanism. Mta2 deacetylates the estrogen receptor alpha and p53 and inhibits their transactivation functions. Mta2 represses the expression of ecadherin in non-small cell lung cancer cells, but stimulates the expression of IL-11 in gastric cancer cells. The Mta2 containing chromatin remodeling complex targets clock BMAL1 complex. Mta2 has been shown to interact with